Okay, we meet again for Sports Nutrition Lecture 2, Hydration, which will probably break into 2 parts. Okay, moving on. These are topics we're going to cover, not necessarily in this order of revised lecture like I do every year. So the order may have changed a little bit, and I've added a couple of new aspects. So homeostasis, a word which should be at least reasonably familiar. Homeostasis is the ability to maintain a relatively stable internal state that persists despite changes in the world outside. So, for example, if you consume sugar, your blood glucose rises, and in most people this triggers production of insulin, allowing effective regulation of said blood sugar. This is of course affected by diabetes. Similarly, during exercise, your muscles increase heat production and your body temperature rises. This is mitigated by sweating. Many of these processes are controlled by negative feedback mechanisms. They occur when physiological conditions change from the normal state and then act to return the body to that state. It's a continuous cycle of events happening, and we can see some examples here. So this homeostatic process in a little more detail using temperature as an example. If the body temperature rises, this is detected by sensors in the skin, leading to activation of temperature regulation systems such as sweating. The second flow chart is a generic view of the process and applies to all such feedback mechanisms, not just biological ones. There's a word there which you may not have come across, effector, the system that brings the body back into equilibrium, in this case the activation of sweat glands. Oh, sorry, that hadn't appeared. It has appeared now. Much of this is controlled by the hypothalamus. This is located below the thalamus, the part of the brain that relays sensory information, and is up a pituitary gland and brain stem as shown here. It's about the size of an almond. Very involved in control of homeostatic systems. Just a reminder, circadian rhythms refers to 24-hour bodily cycles. Okay, so water in the body. As was once noted on Star Trek The Next Generation, we are bags of mostly water, even the brain. Actually, it was ugly bags of mostly water, if I recall correctly. Water is by far the most important biological molecule, as well as roles in the microstructure of biological molecules and many larger scale roles as indicated here. Women tend to have slightly more body fat than men as a proportion, which is linked to childbearing, of course. We can portion the water in our bodies in a number of ways, including whether water is inside cells, intracellular, or between cells, intercellular. Intercellular water is often referred to as extracellular water, and they'll probably be both interchangeably for the rest of this presentation, and particularly in the literature. If you want to do more reading on this, refer to the Total Body Water link, which takes you to the Science Direct collection of topics associated with this. Again, a general summary of the we are bags of mostly water observation. Fat storage is the main source of individual variation, which isn't very surprising. Lipids are very hydrophobic, and adipose cells contain a lot less water relative to other cells, as we can see by this image. Most of the cell is around the edge, with a big globule of lipids in the middle. As mentioned, intercellular water is often referred to as extracellular water. Although, for example, this image refers to 19 litres of extracellular fluids, this is not a continuous system but a collection of compartments, virtual compartments if you will. ICF refers to fluids within the cell membranes and includes water, electrolytes and proteins. Potassium, magnesium and phosphate are the 3 most common electrolytes in the ICF. We have a lot of cells in our body, which are all distinct from each other, so again, ICF is a virtual component rather than something contiguous. Okay, as noted, extracellular fluid exists between cells and is also, of course, the main component of plasma, and probably lymph as well, I assume. It has many roles, including the transport of nutrients. Okay, electrolytes, a little bit of revision from first year chemistry. Fundamentally, an electrolyte is a substance which conducts electricity when dissolved in water. It has specific meanings in biology and particularly sports nutrition. A solution of sodium chloride will conduct electricity, for example. In biology, an electrolyte or a salt will have dissociated into the sodium cations, which have a positive charge, and chloride anions, a negative charge, and these will be surrounded as shown by water molecules. They can conduct electrical impulses in the body and have many other important roles. These are the main electrolytes from the point of view of biochemistry, in particular sports, presented as lists of cations and anions. Remember, cations have a positive charge, anions have a negative charge. Sodium is a main ion lost in sweat. Again, sometimes referred to as an electrolyte. And its deficiency leads to a number of issues which will affect sporting performance as well as general health. Hyponatremia is low sodium concentration in the blood. It is generally defined as a sodium concentration of less than 135 millimoles per litre of blood. There's another definition there. Euhydration is a process of returning to a normal level of hydration. Given sodium losses, they can't be achieved by water alone. Potassium is a key ion in the intercellular fluid. 98% of the body potassium in fluid is in the intracellular fluid, and only 2% in the extracellular fluid. There is some limited evidence that potassium can help retain water in the ICF, but that remains to be established. For general background on potassium, see this link to internal potassium balance. Magnesium is an interesting one. Many sports products promote magnesium, but actually very little is lost in sweat, so their utility is an open question. Magnesium is a good thing nutritionally, of course, with many roles, not least in the action of kinase enzymes and ATP. But is it needed in a sports drink? That's an interesting question. Okay, so just a table showing a differential distribution of ions between ECF and ICF. As mentioned, homeostasis acts to maintain these balances, which will be impacted on by exercise. It's important to maintain these, to maintain electrochemical gradients across and between cells. Perhaps the one you've done most is in human metabolism. Last year you'll have done electron transport, mitochondrial electron transport, which is a big, important electrochemical gradient. Okay, a concept of osmolality. Osmolality is the measure of how much one substance is dissolved in another substance. The greater concentration of the dissolved substance, the higher osmolality, the higher the osmolality. So very salty water has a higher osmolality than water with just a hint of salt. Now this isn't just a concentration term, it's a little bit more complicated than that. So let's talk about that. So osmolality is a function only of the number of particles and is not related to the molecular size, shape, weight or charge. The SI unit is millimoles per kilogram, but there's the milliosmoles per kilogram unit, which is still widely used. An osmole is 1 mole of particles that contribute to the osmotic pressure of a solution. We don't really need to worry about this further, but if you are interested, you see the link down here. The normal osmolality of extracellular fluid is around 280 to 295 milliosmoles per mole per kilogram. Yeah, and that link there will take you to something on how you calculate this if you're interested. But the key point is it's not just about concentration per se, so the weight of something dissolved, it's about the number of particles, and we'll come back to that a little bit later on. Okay, we'll talk about isotonic drinks a bit later on. They may be defined as having a similar concentration of carbohydrates and electrolytes to your blood. That is, they have a similar osmolality. As noted, osmolality is determined only by the number of particles, typically molecules, of course. And there's the osmolality for a range of different drinks, some which are sports drinks, other which are just normal sort of soft drinks. And there it is for carbohydrates. So if we look at these in terms of plotting one against the other, plot osmolality versus concentration of carbohydrate, we get a straight line, if not a particularly good one. The point is there's quite a complex relationship between these factors. So consider this. The carbohydrate in a drink can be small molecules such as glucose or larger molecules such as maltodextrin. You might want to look at what maltodextrin is. The more of the latter, the higher the carbohydrate concentration in terms of weight, but this does not affect the osmolality and the isotropic characteristics of the drink in an easily predictable way. Okay, we'll pause that one now, so we'll come back for part B in a second.